Good morning, everyone. I'm Ananta Chandrakasan, MIT's provost. I'd like to start off by thanking President Kornbluth for her extraordinary leadership for envisioning and driving the institute-wide initiatives, including HEALS. So thank you, Sally. I'd also like to thank Professor Angela Kohler for the terrific job she's done spearheading HEALS over the last year. I'd like to also add my thanks to Professor Ian Cheeseman, Professor Katerina Rebeck, and Caroline Lowenthal for their amazing contribution. Let's give them a round of applause. And of course, this requires a whole team, so I want to thank the entire HEALS team, the Office of Innovation and Strategy team, both for the remarkable work that they've done to get HEALS to this point and for organizing what promises to be a dynamic, fascinating day of discussions. This morning, I have the distinct pleasure of introducing Dr. Noubar Afeyan, who in this room needs no introduction. Noubar is accomplished in so many ways. He's a researcher and inventor. He holds more than 100 patents and is a member of the National Academy of Engineering. He's a business leader, the CEO of Flagship Pioneering, a company based in Kendall Square that creates bioplatform companies to transform human health and sustainability. He's an entrepreneur. He founded Flagship Pioneering and co-founded Moderna, a recipient of the National Medal of Technology and Innovation. He's been a founding team member, director, and investor in a number of other successful ventures. He's an amazing teacher. He lectured at MIT Sloan from 2000 to 2016. He's a philanthropist, a trustee of the Afeyan Foundation. And he deeply cares about supporting and strengthening scientific discovery. Noubar knows as well as anyone the power of science to make a better, safer, healthier world. And he uses his voice to mount a compelling, urgent defense of the value of science, as he did in a recent annual letter for Flagship Pioneering. Noubar is also a long-standing, dedicated member of the MIT community. He completed his PhD in biochemical engineering at MIT in 1987. When the biotechnology industry was still nascent, he was already a pioneer then. Today, Noubar is a member of the MIT Corporation and a member of its executive committee, continuing his wonderful service to the Institute. We've been extremely fortunate that Noubar has lent his strong support to MIT HEALS, agreeing to serve as the inaugural chair of the HEALS External Advisory Council. He's also generously supporting HEALS graduate fellowships that you heard about from Angela, for which we're deeply grateful. And as you've already heard this morning, and we'll hear more as the day continues, the theme of this symposium is moving from discovery to leadership and translation, policy, and ecosystem building. How can we turn breakthrough ideas into solutions that can create real change at a global scale? How can we rapidly improve technologies and use technologies like quantum computing and sensing and AI to solve problems that have been intractable until now, and to accelerate new diagnostics and therapeutics? How can we better connect the research enterprise with practitioners, for example, equipping researchers with patient data to help them understand and tackle real-world problems? On these kinds of questions, I honestly cannot think of anyone better suited to frame the day's conversation than Noubar. He has been the cycle of discovery, invention, innovation, and deployment so many times over and over. We can learn a lot from his experience and insights. Please join me in welcoming our keynote speaker, Dr. Noubar Afeyan.
Okay, it's downhill from here. Well, thanks, Anantha. Thanks, Angela, for doing a phenomenal job getting this off the ground. I don't know how hard it is to to create new things, let alone make them gather this kind of speed. And thank you, Sally, as well. I must say, I was sitting here listening to this long description. I think the one thing you should take away is that people who come to MIT, as I did 43 years ago, essentially have attention deficit order. It's not quite a disorder. If it was a disorder, you probably wouldn't do well here. But the order part of it is pretty close, and that is you tend to learn how to do lots of different things, and if you don't fail at all of them, you keep adding more. I was, as Anantha mentioned, I came to MIT in 1983 as a chemical engineering undergraduate, fascinated by biology at a time when chemical engineers were being allowed to use the word biology. Almost for the first time. And I came here largely because I thought this place had the best in engineering as well as the best in science. And I was interested in the what every bit as much in the how. I figured I could figure out with my engineering skills the how, but the what was not clear to me in biology. And the fusion of those two things that I think you're doing here at Heels is a very important thing. And I'd like to say that, 43 years ago, I caught MIT. It's kind of a disease of sorts, and I have not been able to shake it. There's no real good vaccine for that. And the world needs a lot of people infected with MIT, in my view. And I'm not saying that lightly, I'm saying that pretty significantly. I do think it's a form of a replicating kind of capability. It's in part humility, in part daring, and I think that we play a very significant role and will in the future of health and life science for sure. So I've titled my talk, it used to be called until last night, Defining the Next Era of Health Innovations, and I wanted to emphasize the word unreasonable. So much of what I'm going to say to you is going to sound completely unreasonable, and I want to just make a case for why that should be of interest. And I'll just ask you to pose yourself the question that has been on my mind for a long, long time, and that is simply to say, why do we expect extraordinary outcomes from reasonable people doing reasonable things? And of course, sitting here, you'd say, well, boy, this is the bastion of unreasonableness. Why do you have to say that here? But I would point out to you that individually, we often can be unreasonable. But when groups of us get together, we tend to focus on the common denominator, which is usually reasonable. We ask ourselves, is this going to work? Are these the right people to do it? We do due diligence. We set up committees to review things. These are all things that essentially favor reasonableness over unreasonableness. And yet I would argue, and I'm going to show you kind of a number of examples, that lots of tomorrow's breakthroughs are existent already today in unreasonable form. It's just that we collectively choose to ignore them or underfund them because they're not reasonable. So just think about that as we go to say, where should we put philanthropic resources? What role does the government have, which I'll come back to at the end? I think the government should only fund things that begin, not only, but in the extreme end, unreasonable things. And that I want to just say one other thing. I have lots of slides that I'll skip through, but let me just make one emphatic point. Often in these discussions where there's a strong defense of basic research, and I must say, just this is my own personal view, that I've always struggled with the need to insist that basic research should be pointless, aimless, goalless, that the purest form is curiosity-driven, and if you, for instance, for a moment think of what it could be useful for, somehow it's not basic research. I believe that use-based basic research, that's something that your mind can imagine would be impactful to the planet, to the climate, to agriculture, to just make something up as a motivation to do it, it's the same basic research. But people who have resources will likely relate to that much more so than the artistry that goes into saying, you know what, I don't want to know what it's going to be useful for. So I will make a point as an engineer turned scientist, which is what I consider myself, that it's okay to imagine uses long before you imagine the breakthrough solutions, if only to attract collective resources to the problem. I know this is going to fall on deaf ears, not maybe in this room, but in lots of academic kind of centers. But I would argue that that is one and the same thing. Nothing we do here is basically applied. A lot of it is pretty basic, as you're going to see in a minute. But we motivate ourselves, otherwise known as kid ourselves, into thinking that it's going to have a big impact so that we can actually get enough continuity and kind of, let's say, persistence enough to actually make the breakthrough. So with that as a bit of an off-script thing, let me just tell you a few things that might kind of at least motivate the discussion today. So, you know, I would argue, just in the moment of time we're in, that we're in this interesting transition, and we've been in it for a little while, where we're going from largely an engineered and a discovered world to a world that I think will be increasingly generated. And I think that a lot of the nature, a lot of the things we see around us that was a given before we understood them, will largely be complemented by a lot of things that will be generated by similarly complex processes that humans have not really been able to wield before because we never understood any of them. And so as new, I would argue, forms of complex matter add to the things we don't understand, it's going to be an interesting fusion between what is natural before generative capabilities and natural now, and I think that's okay. In other words, it's humans that are going to basically meld with the ability to create things that used to seem unimaginable, but then we're going to have to make sure that we can deal with those every bit as much as we deal with nature today, carefully, humbly, because we don't understand much of it, etc. So there's a very long a line of research science that's going to have to happen at MIT for a very long time, understanding generated systems, let alone natural systems, in my view. So this is good news for lifetime employment for the scientific part, in addition to the engineering part, in my view. So the next thing I'd like to just say to you is that biology is it. So basically, you know, when I was an engineer and I was very much focused on chemical processes, etc., I thought, boy, biology is fascinating because it's a pre-engineered chemical system. And the reason I say biology is it is because biology is IT. It's just information technology. And we generally haven't thought of it that way. Probably with the human genome sequence and other sequences, we began to at least recognize its information. And we've known long in the central dogma that there's information processing, but we couldn't really say what the hardware is, the software is, how it works. Professor Sharp is here. He made a big contribution to part of the way that processing happens, but that was a major revelation. But then if you think back, we're going to have to figure out all of the information processing to figure out how to tap into that IT part of this. But what is interesting is that it also means that the more we can generate data and start using information technology tools, machine learning tools, and compute, the more I think we'll be able to generate, again, as I mentioned, lots of interesting things. So I do believe we're at this interesting point where I believe that biology will require access to and confidence in and comfort with computational capability. I really don't think, now we could have classical biology where people vehemently stay away from that, but I think that modern biology is going to be very much integrated into an information technology rich field. And you might say, what's the big deal? It's already happening. It is happening, but it's happening with, in my view, some resistance and reluctance, because it almost feels like since we can't figure all this out with IT, then we shouldn't kind of be overly reliant on it. And yet, my guess is that over time, this is my prediction, I think it's going to be a big, big part of this. Now, how does this relate to the industry, particularly the biopharmaceutical industry that I work in? And the answer there is that with AI, and I'm starting with AI because all kind of current talks have to at least make some connection, and I'll come back to it. Basically, you have a convergence happening now between the simultaneous proliferation of all sorts of multimodal measurements, what we call multi-omics in our field, and in the computational side, they look at multimodal. And it turns out that from an information science standpoint, that multimodality informs in very different ways than more data of the same type. And so this richness of data type, you're beginning to see that, and basically this chart's showing the movement from seeing to reading to writing to now beginning to make new stuff, new materials, and new biological systems. And so you're going to hear a lot more about that. The other point that I would just want to make briefly is that if I look on the left side, I do think that discovery in life science has been rather a fragmented thing. People have wanted to go after specific problems, specific capabilities, and so people have made innovations to enable specific things. I remember literally to the day when PCR was initially discovered by Kary Mullis, driving his car. There's a famous story about that. Very soon after, because I was in the field at the time, you could hear the excitement. This is going to be this massive new capability. 50 such capabilities later, we're still finding point solutions to point capabilities and putting pieces together. I think we're going to eventually, and Heels and MIT and others can certainly lead us this way, we're going to have to think about how to integrate many, many capabilities to go after this problem of simultaneously making useful things, but also gaining enough information to be able to direct all this research. But where I've shown here, you're going to hear today about early intervention. You're going to hear about big, bold new biological insights. You're going to hear about superintelligence in science when it comes to coming up with completely new ways to tackle gigantic data. All of this happening in isolation, as in the left, is going to leave a lot to be desired. I think the integration, the proximity catalysis part of this is going to be a hugely important thing. So a lot more room. I remember when Eric Lander with colleagues wrote this paper to motivate the human genome sequence as to what comes after. We're going to need lots of papers that are going to have to envision 10, 20 years from now, however scary it feels, because it feels like we don't even know what's going to happen in 2 years, let alone 10, 20 years. But I think we're going to have to describe those to motivate this integrative approach as opposed to the point solutions. The point solutions will continue. Okay, so as Angela set up very nicely, we live, my firm Flagship lives in this world of asking what if. And what if is our way of making up a use case to motivate a scientific advance that then fulfills a use case. Maybe the same, maybe another one. But that's how we have come to think for the last 25, 26 years. And so we ask ourselves a number of these questions constantly. What if we could see disease much earlier and when it's pre-symptomatic? What if we could reveal completely new chunks of human biology and therefore a richness of new targets? There's always this debate. There's known targets, and then there's hard to target targets. But how to drug targets. But the reality is, it turns out there's a hell of a lot more biology and actual players that we just aren't aware of. So what if we could know those? What if we could leverage the best that nature is doing in genomic engineering? We saw bits of it with CRISPR, but nature almost hardly ever does one thing in one way. There's usually many, many ways. And in fact, we've systematically searched for many other ways genomic engineering is done as nature, and there is a richness of non-CRISPR genome editing, writing capabilities that's there for the taking. And so those are some of the questions. I can go on more. The point is that these each, so forgive me to use this word as an engineer scientist at the top, these each, when they're started, sound like miracles, and therefore people don't believe them. And I would argue that the business we're all collectively in is human-made miracles. If you're the beneficiary of a CAR T therapy, if you're the beneficiary of mRNA that in a vaccine form can actually treat your cancer so much so that it becomes a cure, you think that's a miracle. But those miracles, we all know, come from someplace. And it comes from, in my view, unreasonable people incrementally putting together unreasonable stories, and over time, gathering enough data that reason follows, and the skepticism falls by the wayside based on data. So the burden of the unreasonable is to produce the data. It is not to question their unreasonableness, in my humble view. And this is a place that has to keep that duality, and it has for a long time. I think sometimes we kind of veer towards being overly attentive to what others are doing and how do we do it slightly better. I think we should just do whatever we think is going to produce these human-made miracles. And I'd say that humbly. Obviously, I don't mean the other kind of miracle, but boy, does it feel the same if something unexpected comes forward. So within Flagship is basically a new form of kind of R&D institution that we have developed a number of different advances in novel biology, which I'll give you a couple of glimpses of, novel modalities for new drug forms, AI to make much better molecules, and then AI to completely rethink the scientific discovery process. And I'm going to attempt, as usually MIT folks do, which is to pack a lot in a short period of time, to kind of skim the surface, which I'll pre-warn you will seem not detailed enough to be impressive, but broad enough to be at least intriguing. And any of you who are interested, we're happy to follow up and tell you more. But these are all things that I'm drawing from a wide range of examples and companies that are all within a mile from here. So just to give you a sense, Flagship as an institution comprises 45 distinct companies, all platforms that were developed in our labs at Flagship. Just down the street from here, there's about 10,000 people who work across them today. There's about 50 clinical trials of different drugs and vaccines, and about 150 preclinical programs. I have the distinct kind of advantage of being able to integrate and bring to you little glimpses of these. So this is by no means a single company's work or a single team's work. It's a long, long list of people. Let me just also say that AI, just in passing, is not a new fad within our world. So obviously, biology and bioinformatics has been a lead user of machine learning for quite a long time. Much of what we've done over the years has been pushing the frontiers of big data, models based on big data. And so it's no wonder then that we started one of the very first protein design companies in this space, Generate Biomedicines, that I'll touch on in passing. But then about a dozen of these companies started, all predicated on the use of AI, either to do model-based kind of research as opposed to just more empirical or somewhat less AI-driven models, or generative work of creating new molecules. So I'm going to touch on that as well. So our world kind of falls into these categories. There's a number of things we do in small molecule innovation, large molecules, delivery technologies, genetic medicines, and then on the left side are all these biology kind of forays, and then underneath, underpinning all this is a number of technology platforms, mostly information technology platforms. And the one thing I'll say in passing as well, and this is the interesting thing with MIT, is that MIT doesn't have a medical school, and therefore people kind of feel like it's a bit removed from the end application. I would say that in fact it's in a great position to partner with end applications, plural, because if you are MD Anderson and you're focused on cancer, a lot of these things might be more useful for osteoporosis or for the neurological diseases. And so then if you move back and say, well, what if I'm partnered with MGH, which we are, and many others, MGB, I think what we need to do is think of ourselves as enabling of the entire drug development, drug discovery, as well as obviously device, et cetera, industries, as opposed to try to get very good at one disease area. So what I'm showing in the form of what Flagship does is the number of different entities that essentially are touching different disease states. So to really do this for real, you need to have expertise, enough expertise in all of these areas within HEALS, enough to be able to interact with the broader hospital research ecosystem. So I'm going to take you through a couple of what-if examples and kind of give you little glimpses of what's possible moving ahead. Again, if they seem like no way, is that real? That's fine. If they feel like, oh, I already knew about this, well, then that means you're in the cutting edge of this area. So let me just go through this very briefly. So all of you will know that Moderna did a lot of work over the last 15 years now in enabling mRNA as a molecule to be less immune stimulative, so innate immune stimulative, with chemical modifications, enough that you could package it in LNP and deliver it for various uses. In particular, what became more known was the vaccine use, infectious disease vaccine use. But what is less appreciated is that in parallel, really since 2017, we've been working on one aspect of the technology, which was, could we take a patient's tumor biopsy, sequence using informatics and essentially pattern recognition, identify neoantigens, package those together into a 30, 40, 50 antigen single piece of mRNA, put it into LNP, and cause a very potent immune response to that patient's tumor. And when that started out, it sounded like science fiction, which it was, proudly. And we just needed to say, but what if you could do that? And if you could do that, you could imagine that it would be far more specific to the patient. You might argue, what's the business model? How are you going to create a big biotech factory to make one patient's worth of drug? On and on and on. We had to navigate through that, but I can tell you today that we're way down a path of doing that. In fact, one of the interesting historic things, artifacts, is that because we had gone way down a path already in humans, beginning to test this by 2020, when COVID came, the very process we used to make eventually 1 billion doses, but soon enough 10 million or 1 million doses, was because we had a small manufacturing train that already was taking tumor biopsies, identifying the sequence, converting it into a piece of mRNA, putting it in LNP, and giving it to people. Now, we were doing them one at a time, and we had treated a couple of hundred people, and now we were kind of facing the daunting challenge of saying, substitute cancer with a virus. At the moment, just assume it's one sequence. Eventually, we had to change it over time. But do the rest, except do it over and over and over again. So copy, paste, repeat. So that's an interesting, kind of odd thing in that the one-at-a-time version was more advanced than the things we'd been doing for rare diseases, et cetera. So anyway, that was an interesting coincidence. To make this very, very short, we have a program that we've described publicly, and a product that's in phase 3 trials, nearing its completion, called intismeran, that in melanoma, essentially this is laying out how it works, does exactly what I said, individualized neoantigen therapy. You might say, why aren't we calling this a vaccine? Well, one, you could make up the answer pretty quickly. We weren't doing it for already a year and a half. But two, essentially, it really is a therapy. People tend to kind of forget that if you have a condition, stimulating the immune response to that condition is a really useful thing. It's just not a prophylactic vaccine. It's a therapeutic vaccine. We realize that it's really a therapeutic. It's an immune-modulated therapeutic, and that's what we're working on. And what's been interesting is one piece of quick data I'll show is this was the phase 2 results that showed that in advanced melanoma, you essentially can show a 50% improvement in disease-free survival and in progression compared to Keytruda, which is the gold standard in that case. So Keytruda improves things by about 50%. This improves things by another 50%. And we just released about a month ago, I didn't have the data for that, data that shows that looking at things now over a 3-year time frame and increasingly now 5-year data, we're kind of showing the same effect persisting. So 5 years in cancer is considered kind of, I wouldn't say cure, but as close to cure as people believe in, and we're beginning to get to that kind of data. We have 5-year data, and we're hoping by the end of this year in our phase 3 trials we'll be able to show the same. These were all from phase 2 data, long-term follow-up. What you also can see is that an information technology approach, meaning every molecule is another information molecule, allows you not only to take the same approach, not the same molecule, the same approach, and go after many, many different kinds of tumors, as we have done. Several of them now in phase 3, 3 of them, many, many more behind in phase 2. This is with our partner Merck. But then also we have a number of other approaches with shared antigens, not individualized, that are advancing. So everything you see here is first in category, first of its kind approach to use a technology, synthetic mRNA modified in LNPs delivered through intramuscular injection, same technology, but going after all sorts of different uses in a programmably modifiable way. All right, so let me just change gears. That was a little more detailed. The other ones will be even more skimming, but just to give you variety. Project called Profound, which basically set out to ask the question, what if there's a whole lot of human proteins that we have not previously recognized exist? You might say, how could it be that in 2021, when we started this, there could be proteins we didn't know about? We did the genome sequence a long time ago, and so it's all known. And the reality is, generally in biology, we define things in a particular way, and everything that doesn't fit that definition is called junk. We saw that, by the way, in the 1990s. You may remember at the end of the 1990s when the human genome sequence was done. Famously, Jim Collins basically said that this is like, why would we spend billions and billions of dollars trying to build Bill Hazeltine, even more at the time, well-known people, said this was all junk DNA. I used to go around back in the 90s, late 90s, saying, well, anybody who wants to take their junk DNA out of their genome and see how it goes, you know, the more than welcome to. Junk is whatever we don't understand. It's kind of it's a philosophical concept. But anyway, so there's a lot of that we thought. And so what this project has done is essentially asserted in the what if category that if you could see a mass spec trace with peptides that don't correspond to gene coding regions, of which there are thousands, well known in the mass spec field, and if you could see a ribosome attached to a piece of RNA that corresponds to that sequence, then chances are that's a translated large peptide, small protein, and it doesn't fit the canonical, you know, mRNA with the poly-A and the tail. All of those things are definitional things. Why do I say all this? Well, the team set out to find these. And what's been interesting is that they have found literally tens of thousands of open reading frames of non-canonically expressed proteins, many of them coming from non-coding regions, many of them. So anytime you do a GWAS study and you see something that is hitting a non-coding region and say, damn, I wish it was hitting the actual gene that's doing something, and then you say, it must be a control region, a lot of those are expressed in the form of some form of polypeptide. And now you got to figure out, if you're a biologist, is it repeatedly turned on, turned off in association with disease, and can you target it? Back in the day, that's what we used to call proteins, and that's what we used to go after. So that's what this project is all about. By the way, really doing this without the tools and capability of handling this much data, massive data, without knowing exactly what you're looking for, would be impossible. AI came along in just the right time for this project. And I'll give you one instance of what use you could imagine and what use we used to pretend. We say, look, ADCs are a well-known form of drug now, antibody-drug conjugates. And what if we could find 20, 10, 5 new ADC targets that nobody knew existed. We have found several hundred that we've validated in tumor lines specific. Not all of them we've made any ADCs for, but a whole bunch we have, about 20 we have. This is just showing one such ADC in prostate cancer, very specific, turned on, much more than PSMA, much more specific in our hands at least, it's not in humans yet, which with an ADC we can now drug. Except there's 20 more. And we're not going to be able to, as a single little company, go after these. But you could imagine now that a company like this, or Heals, if it's working in these areas, can start doing partnerships. Now, pharma knows where to go from there. You don't have to do all the way to the drug development. But if you can't connect your crazy idea that I think there's some proteins we don't know about, to here's a few examples and here's what you could do with it, that's the connection that Profound has made in that space. Let me go on. I'm going to give you 3 or 4 other examples and then kind of abstract a bit to close. I've got 9 minutes left in case people are tracking me. So another completely different thing, a project called Biologics. What was that about? Our team wondered, what if you could create a completely new class of protein so that would have advantages over the current proteins that we're used to using as drugs? So you might say, well, what exactly does that mean? And if you just ask that question in a what-if way, you think of lots of different things it could mean. Because immediately you would think, well, I'm going to use unnatural amino acids, I'm going to do tricks with this or that. People have kind of tried to do things in these areas. Pete Schultz and others had kind of worked on unnatural amino acids. This team in particular put forward the notion to one of their what-ifs that, you know, what if you could make an all-D amino acid protein, small protein or large peptide, all D? What would the consequence of that be? Well, a lot of people in this room, if not everybody, could immediately say, well, either I have no idea why you do that, the L1 is just fine, that's the reasonable response. The other thing you could say is, well, an all-D would not be degradable by proteases. Last I checked, there is no natural L amino acid protease that has been evolved to go after a D amino acid polypeptide. So we surmised that. There wasn't data that showed that. There is data that if you just put a few D amino acids in an otherwise L polypeptide, you can affect proteolytic degradation. But an all D, we said, can't touch it if you're a protease. Speculation, by the way. Second, how are you going to present it to the immune system? How are you going to do class switching to make IgG? You got to get it to fit MHC. How's an all D peptide going to fit an all L MHC? Interesting, right? Now notice, we could guess all those things long before we did a single experiment. If you're a scientist, you'd be taught to not do that, because that's speculative, and first do the experiment. But the latter, or the former in this case, motivated doing the experiment. Otherwise, you'd be saying, why am I making all D proteins and peptides? Now, before you think there's a panacea, we went into it. As usual, we always convince ourselves it's a panacea, and then it's not. So we know that. The good news is we know that. It's like exercise. You lose a little bit of weight, and then you get stuck at a given weight. That's the same thing in science. Like the early part gets easy, and then you stop. Where do you stop in this? How are you going to do the screening? You can't make billion compound D libraries because you're not using biology to make these things. So you got to make them synthetically one at a time. So then you say, AI. I'm going to use AI and AlphaFold to be able to predict how an all-D protein fits an all-D target. Answer? AlphaFold only works with all L. All the data is a natural set of data. Has no idea what D polypeptides, which by the way are the same sequence but completely different structure because of the way the chirality works. So the team said, all right, well, we don't know. Let's try. So they basically, by brute force, came up with a few examples of where they could screen for D peptides that would bind to an L protein. And using the first few examples, trained AI models that would then come up with more projected examples, low hit rate, then made those. A subset of those worked. And a few generations later, and probably a year and a half of work later, where we thought, touch a go, if it doesn't work, we don't tell the world anything about it. If it works, great. They now have an out-of-the-gate 10, 15% hit rate computationally to make a potent binding T-peptide against a number of different targets, which is exciting. So we could make up to 50 mer peptides. This is a company called Biologics. We call these synteins. Obviously, you can do the synthesis in an automated way, and it'll be exciting to see how this works. One minor, on the left side, you'll see the use cases that are already being followed and examples being pursued, but on the right side, you'll see one little example, which is tumor penetration. So if you could make a highly targeted all D polypeptide and put it into a use case with a tumor, turns out because of its size, but because also of its persistence, it may have very different properties. And what we've seen is that at least in a few cases, again, this is animal work so far, it's promising. So there's another area. I'm going to skip ahead because my time is running out. We've done these types of things in the gene writing area I referred to. I refer you to a project called Tessera, where we've been using a bunch of different approaches to genome writing, not just editing, that could make a big difference. We have just entered the clinic in AATD, alpha-1 antitrypsin deficiency, with a single one-shot cure. We've shown also that in sickle cell disease, we can deliver LMPs in a targeted way to hematopoietic stem cells. That's entering the clinic within the next 6 to 9 months, we hope. We've got a program in CAR-T, in vivo, again, targeted LMPs that can deliver a single shot modification, very target-specific way. So we've got a lot of exciting stuff in that area. I wish I had more time, but I want to save a little bit of time on the more general comments at the end. Another project called Quotient is looking at somatic mutations in humans as a source of genetic validation of new targets. So the one-liner on that is, it turns out every cell in your body, pretty much, that's dividing, has significant somatic variability between each other. We estimate roughly 10,000 bases different per cell, which means that your genome, on average, is entirely rewritten in your body across the trillion cells you have. So then the question is, where there's variation, I mean, this is probably a Confucius-like statement that Darwin probably made, where there's variation, there's selection. And it turns out that, we know this in cancer, that's what's happening, but it turns out we thought that other than in cancer, variation of our underlying genome, our inherited genome, doesn't really do much. Well, guess what? This team went and posited that if there's variation, there's selection, and therefore if you go into samples that are certain liver diseases, certain cancers, certain different conditions, and deep sequence, millions of cells per patient, you may find enriched mutations that are acquired, not inherited from your parents, that can signal very strongly either a resistant mutant or a helpful mutant to the disease. And in fact, we're finding all sorts of, within 10 patients and 10 controls, you can do the job of a GWAS study with tens of thousands of people. You don't have to take my word for it. There's stuff that they've already published. And this is showing in disease versus healthy tissue, you can literally take zones which have none of the particular manifestation that you're looking for. You take those cells, you sequence them, you compare them genomically, you look at the specific marks. If there's deep penetrance, just to give you an example, in liver disease, in certain liver failures, we've found 6–7% acquired mutations in people with the disease versus not, that are not inherited. And those statistically, with like lots of zeros before a number in p-value, says that that is a gene that's involved. You can't get that from GWAS, at least not so far. Let me skip ahead. Boy, I wish I could tell you a lot of targeted stuff in LMPs. I'll just mention in passing, just so you're aware of it, because this is a company that also has a kind of hereditary connection to MIT, if the least, and hopefully over time we can do more together. There's a company called Lila that we started 3 years ago that set out to use all aspects of agentic and generative AI to essentially take the wheel of science, I would argue, maybe the most valuable wheel humans have invented, although I'm sure people in caves would have argued that the other wheel is also very important. But I'd say the wheel of science, and that is this kind of hypothesis generation, experimental design, running things, modeling, or at least coming up with a way to interpret, and then repeating that repeat, rinse, wash kind of cycle, is what's produced much of the science that we enjoy the benefits of today, and that we think can be done in many, if not all ways, using elements of agentic and AI systems. And we've put a lot of these things together, and we've shown examples across a number of different fields. I'm not going to have time to go through it, but I'd say pay attention to it. There's a company that's about 3 blocks away from here. It's 250 scientists, engineers. We have about $550 million that we assembled to start the project. Hopefully there'll be several more billion that'll be spent creating a whole nother way of doing autonomous scientific discovery. May sound crazy, ambitious, unlikely to work, etc., but our goal is nothing short of creating an automated scientific factory that is essentially directed by humans as to the areas we want to create thousands of hypotheses in, but then the actual generation of experiments and the iteration is done many, many cycles before a human then interacts with what do you do next. I'm sure many of you are having allergic reactions saying, how do you make sure it's safeguarded? And we can talk about that, it's important, but it's probably not a reason not to do it, let alone competitively. Other people won't hold back because the intellectual advance this could potentially, or knowledge advance this could potentially give us is quite remarkable. So more to come there. These are the areas. So in DNA, RNA, proteins, we're way down a path in materials. A number of chemical synthesis materials were also, hydrogen generation, et cetera, we're also advancing. So let me, in just a couple of more minutes, Anantha, if I can go just a couple more minutes late, end with saying a few things more generally. So thanks for hanging on to the roller coaster of the first part. So as Anantha said, one of the things, having done this for now 39 years after graduating from this place, still afflicted by MIT, I kind of feel like at no time has the underlying process of science been remotely as endangered as right now. And so I wrote a paper with some collaborators and put it out in the form of a letter called Choosing Science, and it makes basically the following points, which is that on the one hand, the pace and breadth of human-made miracles on which we're relying for human health, for agriculture, sustainability, climate, many other things, is increasing, proving itself, proving its economic worth, and is poised to increase even more. But the underlying assumption of that is that you actually believe what it generates, and that you believe that in order to debate things in science, you have to have facts against facts, not facts against opinion. Now, ironically, in science, we train students to be skeptical. Long ago I heard science described as just organized skepticism. And so everybody is skeptical about everything somebody else does, and so collectively we're skeptical about everything. Often there's always a special, a little pocket reserved for lack of skepticism in what one does themselves, unless they're really elite, but most of the time they're very happy shooting down other people's ideas. That's a good thing in science. But you're doing so with science, with data and facts going up against others, not opinion. But the problem is now in the public discourse, and particularly as a result, I think, of COVID, where in the heat of the exigent moment and thousands of people dying and us all seeing body bags, people made proclamations about the certainty of science that caused other people to say, wait a minute, that turned out not to be true, and therefore I can criticize any of it, in fact, all of it. And they've started with, obviously, vaccines and then mRNA, but now autism and many other areas. And the problem with that, to me, I'm not up to defend it against any one thing. But if you start questioning the scientific method, especially at a time, as I lay out the case, when we have a pretty significant, well-funded competitor in China that is doubling down on the scientific method and the progress that it portends to make advances in electric vehicles and battery storage and quantum and AI, all sorts of fields, it's a pretty rough time to be doing that. So I think that we need to essentially step back and say, it's not okay. Unless you come up with a better way to do fact-finding in science, I think up until then we need to stick to what we know. And as a scientific community, we need to speak up. I'll tell you that a lot of scientists don't speak up except if somebody goes after their science. That's a mistake. We should speak up when they go after our science and equally when they go after the scientific method. Because it's really important that everybody insists that facts be presented and not kind of opinion to guide policy. So that's one thing. I say a little bit here about China. I will not go delve into it. I do think that as a kind of current moment, there are things that we need to be thoughtful about in terms of the competitiveness that a centrally organized, planful economy can offer in biotechnology compared to a US system that is largely, entirely, proudly capitalistic, but has very little kind of analysis being made as to how we're losing competitiveness. We are losing competitiveness. I think we should be allowed to compete fairly, but there's a number of things, policy things that I'll touch on just in passing. We're not going to have time to get into them. One thing I do want to mention is that the biotech industry, which is one of many that are going to be interested in HEALS, is a $3 trillion impact industry today, right? It employs 2.3 million people in 2023. And essentially 60% or more of all new drugs are coming out of the small biotech industry. You cut that off, and that's going to be a major, major problem and challenge, let alone what we just saw in health security. The nation's security was dependent on health security. Health security are based very much on the advances that this industry, whether diagnostic or therapeutic or vaccines, are making. And so I think this is a case that we all need to be familiar with and make. And there's a number of policy recommendations. This is probably not the right forum, but anybody who's interested in this, that we've made publicly, and the Council on Foreign Relations has put out in a report that they've done, and we contributed our elements of this there. So anyway, I'll just mention that if some of you are in the room thinking, I'm just a scientist, I'm just an engineer, this is not my kind of science, this is political science, the answer is when political science impacts your kind of science, you need to be willing to engage in political science. And that is if you just kind of say, well, you know, I don't need to engage with that, funding will suffer, a lot of other things will suffer. And then I'll leave you with just one last thought, and that is this notion of polyintelligence. So a couple of years ago, we put forward this notion, and I'd urge HEALS at some point to think about this, and that is, you know, at a time when we are fascinated at the interface between human intelligence and machine intelligence, and on the other hand, as biologists, as scientists, we've often been fascinated at the interface between human intelligence and nature's intelligence, namely nature. I would argue that what's really more interesting is the triangle that's formed when all three, that is human reasoning, imagination, knowledge gathering, coupled with what we can do with nature's forms. I believe fundamentally that nature is just a whole bunch of intelligences. And we've never viewed it that way. I'll make an impassioned appeal to you that you reconsider that. Humans made up language. We therefore control what intelligence means. And up until recently, we were the only intelligent thing on the planet. So animals were definitely not intelligent. And in any event, machines weren't intelligent until we made this room for artificial intelligence. Once we did that, and once we realized, holy cow, it might be close to this, AGI, that, now we've opened a bit of a Pandora's box, and I'd suggest we have to go back and think, is a virus that almost took down the planet's human immune system intelligent or not intelligent? If you define intelligent as a learning system that is adaptive and that can handle all sorts of forward-looking situations the way humans do by adapting their actions to new information, then you'd say that's a form of intelligence. Everybody's as much as a bamboo tree that flowers every 50 years precisely, or everybody as much as your own immune system that fights cancer every day that you're alive in micro forms. So once we start saying, hey, nature is just a bunch of intelligences, remember my biology is it, now you could say, well, why don't we use artificial or machine intelligence to start emulating nature's intelligences at the level of complexity they both have, both of which absolutely crush the human ability to keep straight 1,000 things and how they're connected. Just like we have no means to do that. Physicists get away, kind of a little bit easily because it turns out that mathematical systems tend to describe these basic principles that even recursively at large scales hold. They stay away from biology, since Schrodinger, for a good reason, is that has not proven the case. I would argue that a man-made augmentation of our intelligence, namely large data models, juxtaposed to nature's intelligences, will allow us to make much more progress than humans with their own means, with a little bit of computing, trying to be fascinated by nature. So you might say, big deal, but I'll just kind of leave to you the thought that this is nothing more than a way to expand our reasoning skills and our imagination skills. And I'll leave you with this last quote, which is, I do think we're entering an era where we're going to have to train students and scientists to realize it's okay to use your imagination, to foresee things, unreasonable things that don't yet exist, and then figure out what experiments to do to show whether they could exist, they should exist, and then make them exist. That's a little different than chasing the next adjacent hypothesis and the next one and the next one, and then when you're endowed, surprised by a major new finding, get rewarded for it. Both have their own justification. I'd say useful things will come out, especially with AI, a lot more with this. So I kind of end with the quote from George Bernard Shaw, imagination as the beginning of creation. You imagine what you desire, you will what you imagine, and at last you create what you will. I think AI will contribute to imagining what we desire, to willing what we imagine in making it a lot more actionable, and then obviously helping us create these things. So thanks for your attention.